Welcome to Tomorrow Unveiled, your weekly shortcut to the future, where we cut through the hype and dive deep into the most significant breakthroughs shaping human potential.
And this week, we are synthesizing what our research team is calling the Immortality Update. And this is not about the philosophy of living forever.
No, not at all.
It's focused on functional life extension. And that's a crucial distinction, isn't it? It's not just about adding more years to your life, but about dramatically extending the decades you live in robust health.
With vitality, with independence. Exactly. For so long, longevity science felt like, well, theoretical biology. It seems stuck in early stage trials or, studies on worms and mice.
Right.
But the data that came in this week shows a really decisive shift. We seem to be moving towards something practical, measurable, and I think most importantly, something modifiable.
We are. The precision we're seeing in the basic research is, well, it's astonishing. We're talking about flipping specific genetic switches or designing AI clocks that can tell you your true biological age.
Not just your birthday.
Exactly. But there's this core tension. It's really defining the field right now. On one hand, you have this almost science fiction level of precision in the lab.
The biological software update in action.
That's a great way to put it. But on the other hand, you run into the friction, the stubborn friction of clinical reality.
A trial failure.
Trial failures, huge questions about who gets access to this, and even major funding collapses that could slam the brakes on all this incredible momentum.
So our mission today on Tomorrow Unveiled is to cut through that noise for you. We've synthesized all the key findings that were strongly corroborated across the source material this week. These are the breakthroughs that multiple teams agree are real and consequential.
This is the deep dive into what happened this past week, November 18th to the 25th. And I think we should start right at the foundation.
Cellular maintenance and rejuvenation. Let's do it. We're starting with the most direct attack on aging, really going after the hallmarks of aging right inside the cell. For a while, the focus was all on senolytics, these drugs that kill off zombie cells.
Right.
But the newest research suggests the body might have a better, a more elegant solution already built in.
Precisely. We're seeing the discovery of the body's own internal system for managing these aging cells. The research team is calling it the senescent surveillance paradigm.
Okay.
Before, the idea was we needed to invent drugs to be the cleanup crew.
Yeah.
But now it turns out the body already has a very sophisticated, sometimes sleepy cleanup crew on the payroll.
So who are these internal assassins? How do they work?
They're a specialized type of immune cell. They've been identified as CD4 EOMES cells. They actually start out as your standard T helper cells, the ones that usually coordinate an immune response.
Right.
But the research shows they go through this crucial phenotypic switch. When they run into a senescent cell, one of those toxic zombies, they turn on a specific gene called EOMES, and that basically transforms them.
It turns them into highly specific killer cells. So the helper becomes a killer, but only when it sees a specific target.
That's the upgrade. It's a key piece of our biological code rewriting itself in real time. Their job is essential. They patrol our tissues and eliminate these senescent cells, which are so damaging because they secrete that inflammatory cocktail.
The SASP.
The SASP, exactly. The senescence-associated secretory phenotype. And that's what drives so much of the chronic low-grade inflammation that accelerates almost every single age-related disease.
Okay, but the source has pointed out a paradox here, which I found fascinating. If our bodies have this internal cleanup crew, why do we accumulate zombie cells as we get older? Why doesn't it just work?
That's the critical question that really moved the science forward this week. It turns out that in older models, the number of these CD4 EOMES cells actually goes up.
So the body knows there's a problem.
It knows. It's trying to compensate. But their efficacy, their actual function, drops off a cliff. They're there, but they're tired, they're poorly deployed, or maybe they're just overwhelmed. Think of it like a security team that's just too slow to respond to 10,000 alarms going off at once.
So the deployment protocol is faulty in old age, not the hardware itself. What does that mean for future therapies then?
Well, the implication is huge for treatment. Instead of just relying on drugs that might have all sorts of off-target side effects, we can start thinking about immunotherapy for aging. We could potentially use something called adoptive cell transfer, or ACT.
ACT, I remember that from cancer research. That's where you take a patient's own cells out, you engineer them, and you put them back in, right?
Exactly. It's being called CAR-RT for aging. You'd harvest a patient's T cells, then outside the body, you use genetic engineering to basically train them to be better killers. You make sure they express EOMES efficiently and that they specifically target the senescent cells.
And then you reinfuse them.
You reinfuse them.
Yeah.
You're restoring the body's own maintenance function with incredible precision, completely bypassing that age-related bug in the system.
That sounds incredibly precise, but we still have to deal with what the research called the Goldilocks problem. It turns out not all senescent cells are the bad guys, are they?
And this is the kind of nuance that separates future medicine from today's blunt instruments. A major review highlighted that some senescent cells act as sentinels, especially in barrier organs like your lungs or your skin. When you get an injury, these sentinels secrete very specific, localized growth factors that wake up nearby stem cells to start the repair process. They're the first responders.
So if we just use a drug that's like a nuclear option, killing every senescent cell everywhere.
You might clear out the inflammatory zombies, yes, but you could also cripple your tissue's ability to repair itself after a cut or a burn. You kill the sentinels too.
So the challenge for what they're calling precision senolytics is to distinguish between the good guys and the bad guys.
Precisely. To develop a therapeutic language that can tell the difference between the repair-inducing sentinels and the degenerative zombies. And this discovery of the CD4 EOMES pathway is giving us the molecular map to start programming that very distinction.
Okay, so moving from the microscopic cleanup crew to therapies that are already in the clinic. The speed at which cell therapies are moving from the lab to humans is one of the most exciting things we track on Tomorrow Unveiled.
And the most successful clinical finding from this week was in a phase 1-2 trial for age-related macular degeneration, AMD, specifically the dry form.
Which is the leading cause of blindness in the elderly, and historically we've only been able to slow it down, not reverse it.
Exactly. The intervention here involved transplanting healthy adult retinal pigment epithelial cells. These are the support cells for your photoreceptors. And the trial showed that this transplant could actually restore sight.
Restore sight. What was the specific data point?
This is the one that jumped out. Participants gained the ability to read an average of 21 additional letters on a standard eye chart after just 1 year.
21 letters. That is not a small improvement. For a lot of people, that's the difference between being legally blind and having functional independent vision.
It's the difference between reading a book and not. And crucially, the safety profile was excellent. No serious inflammation, no rejection, no tumor growth. This is one of the first truly restorative treatments for this kind of functional loss.
That's incredible for a specific organ, but the research also corroborated findings on systemic aging right back at the foundation of our entire body, our blood stem cells.
That's right. Research from multiple top-tier institutions show that you can reverse aging in blood-forming stem cells, or HSCs, by targeting one specific part of the cell's machinery.
And that is?
Lysosomal dysfunction. The lysosome is the cell's garbage disposal and recycling center. And what happens is, in aged stem cells, these lysosomes become hyperacidic and hyperactive. They're basically working overtime, running way too hot.
And that's a bad thing.
It's a very bad thing. It paradoxically reduces their regenerative potential. Your stem cells can't make healthy immune cells, and you get immunosenescence. But by targeting this hyperactivity with specific compounds, researchers were able to restore the health of these fundamental cells.
So did they just look younger, or did they act younger?
They absolutely acted younger. Suppressing that hyperactivity restored blood-forming capacity, it renewed mitochondrial function, the cell's powerhouses, and it balanced out immune cell production. The critical takeaway, as the researchers put it, is that aged cells can indeed bounce back.
Bounce back, I like that. So aging in our blood isn't necessarily a one-way street of damage accumulation.
It's not. They retain the capacity to become youthful again if you fix the underlying hardware fault. And that opens up huge avenues for improving immune response in older patients.
Okay, if that section was about fixing the cell's maintenance crew, this next one is about regulating the cell's engine and energy supply. We're moving into metabolic manipulation, using drugs we already have, and, well, digging into the kernel of our biological software.
Starting with the GLP-1 receptor agonists. This is the class of drugs that includes things like semaglutide. They are, I would argue, the most consequential near-term clinical target in all of longevity science.
Because they're already out there, they're safe, and we're finding out they do much more than just help with weight loss. The research this week detailed some mouse studies that gave a really strong reason why they might work for systemic longevity.
Yes, a research team treated middle-aged mice with a low-dose GLP-1 agonist for 30 weeks. And the key here was the dose was managed specifically so it would not cause weight loss.
So they could separate the anti-aging effects from just the effects of being leaner?
Precisely. And they found the drug produced these strong, body-wide counteracting effects on aging that looked almost identical to the effects of caloric restriction and rapamycin.
Which are the gold standards. So you're getting the benefits of CR without, actually starving yourself. What were the specific functional benefits?
They were broad. Improved grip strength, better motor performance, enhanced cognition. And importantly, this only happened in the aged mice. It didn't do anything for the young adults.
Which confirms it's reversing an age-related decline, not just juicing up performance.
Exactly. And the mechanism they identified was key. The benefits required signaling through the hypothalamus in the brain.
The body's master controller for energy.
Right. So this drug isn't just acting locally in the gut. It's signaling through the brain to coordinate a body-wide shift to a more youthful metabolic state. It's a profound brain-body axis.
It's the brain deciding the system is safe, so it can prioritize long-term maintenance. That's a huge insight. And on the practical side, there was a major development in the U.S. about these drugs.
That's right. Medicare announced substantial price reductions for this class of drugs. And while that's for diabetes and obesity now, it's crucial. It makes them more accessible and, critically, it lays the groundwork for future coverage if they get approved for frailty or longevity. The infrastructure for widespread use is being built right now.
Okay, let's shift from these powerful whole-body drugs to the microscopic gas and brake pedals inside the cell, the genetic regulators.
And we'll start with Sirt6. It's often called the longevity gene. We've known that boosting Sirt6 can extend lifespan in mice, but the question was always how. This week's research seems to have figured that out.
So what's its secret?
Sirt6 acts as a really sophisticated regulator for the production of hydrogen sulfide, HS.
Okay.
HESS is a crucial gasotransmitter, a bit like nitric oxide. It's essential for vascular health, and its production drops off a cliff as we age. Sirt6 doesn't just make it, it fine-tunes it.
Fine-tunes how?
Under stress, Sirt6 hits the gas pedal. It recruits the enzyme that makes HSS to ramp up protective mechanisms. But here's the clever part. It also acts as the brake. It represses that same enzyme when HHS levels get too high and risk becoming toxic.
So it's managing the dose in real time, keeping it in that perfect protective zone.
Exactly. And this dynamic fine-tuning perfectly mimics the benefits of caloric restriction, but without the physiological stress of starvation. It's strong validation that Sirt6 activators are true caloric restriction mimetics.
A much more achievable path for most people. Okay, so that's Sirt6. Now, what about NANOG? The research team that compiled this information called it the Conductor of Metabolism. I always associated NANOG with embryonic stem cells.
And that was the breakthrough. Finding that NANOG, which we thought was just for achieving pluripotency in stem cells, has a crucial active role in our adult cells. It governs what they call a bivalent metabolic state.
Can you break down what that means?
Think of it as metabolic flexibility, plasticity. Most of your adult cells use one primary system for energy, a slow, steady burn called AUXPHOS. But stem cells need to be able to do everything. They need high glycolysis, a rapid sugar burn, and high AUXPHOS to support growth and repair.
And NANOG is the conductor making sure they can switch between those states.
Exactly. As we age, NANOG expression declines, and our cells become metabolically rigid. They get stuck in one gear, and that loss of adaptability is a core feature of aging.
So NANOG is keeping the system flexible, which is really a definition of youthfulness.
Precisely. And on top of that, NANOG was found to protect cells by directly activating a powerful antioxidant enzyme called GPX2. So it's a master regulator for both metabolic flexibility and defense against oxidative stress. Targeting it is a really comprehensive strategy.
This information is dense, it's revolutionary, and frankly, it changes how we think about the human body. We've gone from just slowing things down to understanding the actual software inside the cell, finding these kernel parameters like Sirt6 and NANOG.
It's a complete paradigm shift. We're not just mitigating decline, we're learning the biological language of youth. And that language is proving to be far more modifiable than we ever thought possible.
If you're finding this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future.
And we love hearing your thoughts. Whether you're on YouTube or your favorite podcast app, leave a comment and let us know what you think about this week's longevity update. Which of these targets, like NANOG or Sirt6, do you find the most promising?
And if you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners. Now let's move on to Section 3, Measurement. Because to manage this software update, we first have to measure the system.
And we have to do it accurately and continuously.
Right. This shift to a quantifiable biological age is maybe the biggest change for the average person. Aging used to be this vague philosophical thing. Now technology is turning it into a data problem.
And that's the core of the health span economy. If you can reliably measure your rate of aging in real time, you can optimize everything far more effectively. And this week, we saw the debut of some landmark AI-powered clocks that do exactly that.
Which ones really stood out?
Two in particular, Life Clock and PPGH. These are major algorithms, trained on millions of health records and activity tracker data. Life Clocks, for instance, was trained on over 24 million clinical visits. These tools show that continuous, personalized aging measurement isn't science fiction anymore.
And the one that seems most ready for prime time uses the tech we're already wearing on our wrists, the AI PPG age.
Yes, this is a deep learning algorithm that assesses your vascular age directly from the raw pulse data from your consumer wearable.
Like a Fitbit or an Apple Watch.
Exactly. They all collect these PPG signals. The AI interprets those raw, complex signals to estimate the stiffness and the overall health of your blood vessels.
And why is vascular age such a powerful predictor?
Because your vascular system is often the first thing to show wear and tear from chronic stress. A higher AI PPG age gap, meaning your arteries look older than your calendar age, was found to be a very strong predictor of future cardiovascular events.
So we're talking heart attacks, strokes.
Heart attacks, strokes, onset of type 2 diabetes, and all-cause mortality. It makes it a highly scalable, non-invasive biomarker. You're moving aging assessment from an annual blood test to a continuous, real-time dashboard on your wrist.
So in theory, I could see my AI PPG age jump after a really stressful month, giving me immediate feedback that my body is accelerating its aging.
That's it exactly. It makes the abstract idea that stress is bad into an immediate, actionable data point. It turns aging into a micromanaged data flow.
Okay, that's AI and peripheral measurement. But now AI is going even deeper, using imaging to assess structural aging inside the body. Let's start with that invisible accelerator of aging, chronic stress.
Measuring chronic stress or allostatic load has always been a huge challenge. But the breakthrough here is something called the Adrenal Volume Index, or AVI.
How does the AVI give us an objective measure of stress-induced aging?
An AI model was trained to measure the volume of the adrenal glands from existing CT scans. The biology is simple. Chronic stress makes these glands work overtime, producing cortisol and adrenaline, and that leads to measurable enlargement or hypertrophy.
So a bigger adrenal gland is a physical signature of accumulated stress, a stress age.
Precisely. A higher AVI correlated directly with a higher stress load and lower longevity. It's the first objective, repeatable radiomic clock for biological aging caused by stress. It lets researchers go back through millions of old chest CTs and retrospectively quantify the stress age of entire populations.
And then there was the monumental work on the brain itself, the 5 epochs of the human brain. This wasn't just about measuring decline, it was about mapping the entire operational lifetime of the brain.
A colossal study. It mapped the brain's structural evolution from age 0 to 90. It basically transforms brain aging from this gradual downward slope into a quantifiable epoch-based timeline.
And what were the key transition points they identified?
There were a few, but two are really critical for this conversation. The first happens around age 32. That marks the peak of neural efficiency. The brain then enters what they call the stability plateau.
A plateau.
A long phase of peak function and robust stability that lasts from age 32 all the way to age 66.
34 years of functional stability. That's the target for longevity science, isn't it? Extending that plateau. And what happens after 66?
That's the next big shift. It marks the start of the degeneration epoch.
Yeah.
Myelin breakdown, network reorganization. It all starts to accelerate. And the relevance here for interventions is profound. Meaning treatments aimed at protecting the brain, whether it's a GLP-1 or something else, have to be given during that long stability plateau. Waiting until you have cognitive symptoms when you're already in the degeneration epoch is likely far too late for reversal.
Prevention is the only viable strategy. And to make all this measurement science work, we need a baseline, a control group. That brings us to the Precision Aging Network data release.
And that data set is arguably the most valuable tool that came out this week. It's a massive 300 terabyte database of multi-omic data focused specifically on healthy aging. Most research studies disease, this studies resilience. It's the reference manual for what optimal health looks like at any age.
Okay, we've established the revolutionary science and the incredible precision of the new measurement tools, but now we have to talk about where the rubber meets the road: human clinical trials. And this week gave us a pretty sobering reminder that what works in the lab doesn't always work in people.
The most disappointing news, without a doubt, was the failure of the GLP-1 drug in those two major phase 3 Alzheimer's trials, EVOKE and EVOKE+.
So despite all the promise we just talked about with metabolic rejuvenation in mice, it failed. It didn't slow cognitive decline in humans compared to placebo.
It didn't. And the research team that compiled this information suggests it points to one critical concept. The intervention came too late.
Too late in the disease cascade.
Exactly. If you go back to those 5 epochs of the brain, by the time patients are enrolling in a phase 3 trial, they already have noticeable cognitive symptoms. They are firmly in the degeneration epoch.
So if the brain has already lost synapses and developed significant structural damage over decades, even the best metabolic support can't rebuild what's already gone.
It can't. The biomarker analysis from the trial was actually positive. It showed the drug was having the intended metabolic effect in the brain. The mechanism was valid. But the structural damage was likely irreversible. This failure is an expensive but powerful reinforcement of the need for early preventative action during that stability plateau.
The lesson being that if you want deep structural change, you might need a deeper, more structural tool. Which brings us to gene editing, the ultimate software patch.
And we saw 2 incredibly targeted genetic interventions this week that really moved the needle on major disease risk. It proves that the rare genetic advantages some centenarians have might actually be transferable.
Let's start with the big one, editing the APOE4 gene for Alzheimer's risk.
APOE4 is the single strongest genetic risk factor for late-onset Alzheimer's. If you have 2 copies, your risk could go up by a factor of 10 or more. Researchers successfully edited that dangerous APOE4 gene in living mice, turning it into its protective counterpart, APOE2.
And this was done in a living animal, not just in a petri dish.
In vivo, yes. And the results were fantastic. The edited mice showed significantly less Alzheimer's pathology, less amyloid plaque, less inflammation. And what was really remarkable is that this worked even when the gene was edited later in life.
That's a huge step forward. It suggests you could package the protective genetics of a centenarian as a one-time preventative gene therapy for high-risk people. You're literally rewriting their genetic destiny.
It's profound. And then on the cardiovascular side, there was the work on Serpium-1 reduction. This is the one that reported that dramatic 20% life extension in mice.
So cardiovascular aging is often about tissue stiffness, fibrosis. How does this gene fit in?
The Serpium-1 gene produces a protein called PAI-1, and PAI-1 is it's a bad actor. It actively promotes fibrosis and clotting. It's a well-established pro-aging protein.
So the goal was simple: get rid of PAI-1, and the impact was?
They studied mice that were bred to not have the Serpium-1 gene at all. These mice lived an astonishing 20% longer than their normal litter mates. More importantly, they were almost completely protected against the hallmarks of cardiovascular aging. Their hearts stayed flexible, they resisted hypertension.
This confirms PAI-1 is a major central pro-aging protein. Targeting it is a direct path to slowing down the structural decay of our most critical system.
A 20% life extension from knocking out a single gene is the gold standard in this field. It validates the idea that aging might have just a few of these master switches.
Okay, we've talked cellular mechanics, we've talked genetic code, but our sources also pointed to a really surprising study on Psilocybin, the active compound in magic mushrooms. How does a psychedelic fit into the Immortality update?
It forces us to connect the mental state directly to the cellular state. The study suggests psilocybin has systemic anti-aging effects that go way beyond its known mental health benefits.
What were the specific non-psychological markers that it affected?
In the lab, it extended the cellular lifespan of human skin and lung cells by over 50%. In animals, it improved DNA repair, it reduced oxidative stress, and it helped preserve telomere length.
So it's influencing fundamental hallmarks of aging: genomic instability, telomere attrition.
It is. And the implication here is a kind of psychosomatic view of aging. The idea that neural inputs, specifically through the serotonin receptors that psilocybin targets, can regulate cellular stress responses throughout the entire body. The brain isn't just a victim of aging, it's a commander of the aging process.
So if chronic stress accelerates aging through cortisol, this research suggests a compound that reduces existential anxiety might, at the same time, signal to cells all over the body to enter a protective, repair-focused state.
Exactly. It could be a prototype for a whole new class of geroprotectors, one that bridges the gap between mental and biological health span.
We've covered cellular reversal, metabolic revolution, precise measurement, and gene editing. The scientific acceleration is undeniable, but this momentum exists within a very complicated institutional and economic reality.
And we have to address that. The field is experiencing what you could accurately call a fragile momentum. Despite all these breakthroughs, the sources highlighted some serious friction that threatens to slow things down.
The biggest shock from the industry side was the end of that massive, long-running longevity partnership.
Yes, the dissolution of the AbbVie-Calico partnership. It was a stark reminder that scientific possibility doesn't automatically equal a successful product. This was a billion-dollar, decade-long endeavor. Its collapse shows how long and precarious the path from discovery to medicine really is.
And the risk isn't just from the private sector, it's also coming from the government, which funds all the foundational science.
We saw corroborated warnings of potentially corrosive NIH budget cuts. Some estimates were as high as 40%. The NIH is the lifeblood of basic science. It funds the early career researchers and the big multi-decade studies like the PAN dataset we just talked about.
You cut that foundational funding and the entire pipeline of discovery slows to a trickle.
Immediately.
Which brings us back to the listener's world. Access and cost. If these therapies are so revolutionary, who's going to get them?
The cost barriers today are, well, they're substantial. Longevity clinics can cost anywhere from $15,000 to $250,000 a year. And even crucial repurposed drugs like GLP-1s often aren't covered by Medicare for things like frailty. It creates a two-tiered system right out of the gate.
And the ultimate concern here is the longevity divide.
Experts warn that if these cutting-edge treatments, especially things like gene editing, the APOE4 hack, remain prohibitively expensive, we risk creating a biological caste system, where the wealthy aren't just richer, they're biologically superior and live decades longer in good health.
A shift from economic inequality to biological inequality.
It's a massive societal challenge we have to face now. But the counter-argument is powerful too. The research team highlighted that extending healthy life expectancy by just 1 year is valued at $38 trillion globally.
The health span economy. It's not a boutique market, it's an economic imperative.
Absolutely. Keeping people functional and productive for longer creates far more economic value than the downstream costs of managing chronic disease.
And yet, underlying all of this is the sobering realization of the demographic plateau.
A major study confirmed that the era of rapid life expectancy growth from low-hanging fruit, sanitation, vaccines is pretty much over in wealthy nations. We've hit a hard ceiling around 80 or 85.
So to cross that ceiling, we need radical functional life extension. We need the deep biological engineering we've been talking about.
We do. But let's end this part of the dive on a slightly more accessible note. Systemic interventions that don't rely on expensive gene edits, but on harnessing the biology we already have, specifically in our gut.
Preventative maintenance that's largely actionable today.
Right. And one of the most promising is a compound called urolithin A.
What is it and how does it work?
It's a metabolite. It's produced when certain gut bacteria break down polyphenols from things like pomegranates. And research showed that supplementing with urolithin A in middle-aged adults effectively rejuvenated their aging immune systems.
How does it do that at the cellular level?
It enhances mitophagy, the process of cleaning out damaged mitochondria. It specifically targets T cells, the core of our adaptive immunity. By clearing out that cellular junk, it basically gives the aging immune system a vital refresh.
And finally, the broader idea of microbiome manipulation seems to be getting more and more targeted for longevity.
It is. Studies are now identifying specific microbial profiles linked to exceptional longevity. For instance, long-lived mammals like giant pandas have high levels of bacteria like Akkermansia mucinifila. This points to a strategy where we modulate the microbiome to produce these beneficial compounds for us inside our own bodies.
Turning the gut into a personalized, continuous longevity factory.
Running on simple dietary inputs.
Yeah.
Exactly.
What an incredible week of breakthroughs. To summarize this Immortality update, the path forward really does feel like a software update for the human code. We saw the logic of NANOG, the structural timeline of the brain epochs, the diagnostics of the AI clocks. Aging is a data problem we are finally learning to read.
And we've established that aging is not this inevitable one-way decline. It is highly malleable and, in some cases, clearly reversible. We saw that with the blood stem cells that bounce back and with the reversal of APOE4 risk with a single gene edit.
And the immediate future seems to lie in a combinatorial paradigm, layering these things together. Precision diagnostics like the AI-PPG age, with immune rejuvenation, with metabolic reprogramming. The search for one magic bullet is over. The era of systems engineering the human body is here.
But as we rapidly gain the tools to extend our functional lives, the ultimate question remains unanswered. Are we building a society that can actually support those extra years? Our sources highlighted a truly sobering data point.
Which was?
That suicide rates are highest among older men. And it's often driven by a loss of purpose, social isolation, and the despair that comes with physical decline.
That's a devastating implication. If we solve the biology of the cell but ignore the sociology of a 100-year life, we haven't actually succeeded at all.
Right. Biological longevity without psychological longevity, without purpose, community, mental health support, is a recipe for a future crisis. The true challenge of the $38 trillion health span economy isn't just perfecting a gene edit, it's designing a fulfilling and meaningful 100-year life. And that is the provocative thought we'll leave you with this week on Tomorrow Unveiled.
Thank you for joining us for this deep dive into the Immortality update. We'll be back next week to continue tracking the breakthroughs shaping your future right here on Tomorrow Unveiled.